Okay, thank you guys. I'm sorry for the delay. Um, can everybody hear me on Zoom? We're all set now. I'm gonna assume that's a yes. Okay. So I like to start with this slide when we're talking about biological imaging. It shows us the history from where we're coming from the very first micrographs hand-drawn by some of the greatest scientists and biologists in the beginning of time. And it shows how biology has always been an imaging science, documenting phenomenon by drawing and by taking pictures from the first Leeuwenhoek microscope, which is basically a glass bead, to now today our super duper Titan cryo-electron microscopes. We've gone a long way in about 100 years. So I'm going to talk to you today about biological electron microscopy, workflow, sample prep, cryo-TEM. So my name, if you don't know, is Nicki Watson, and my email nwatson2@fas.harvard.edu. If you want to talk to me later, if you want to have a consultation, if you want to just talk about things that we can do for biological electron microscopy, just contact me. So it all started with electron microscopy with George Emil Palade. In the 50s, he was, when the first electron microscopes came out, and him and his group of people at Rockefeller wanted to use this instrument to help form and create the science of cell biology. And so he won the Nobel Prize in 1974 for work done in the 50s. His work was primarily in imaging-wise, was imaging ribosomes, but also cell fractionation through differential centrifugation. He was, I say, most noted for ribosomes and endoplasmic reticulum, and which he first described in the 50s. He is often described as the most influential cell biologist ever, essentially because before him, the science of cell biology didn't really exist. The same preparation methods developed by Palade and his group at Rockefeller are the same methods that we're using today for classical electron microscopy. So this is also an example of one of the very first electron micrographs of a whole cell done at the Rockefeller Institute. If you look closely, you can see the mitochondria very clearly. You can see ribosomes very clearly. You can see the endoplasmic reticulum, if you know what you're looking for, actin bundles, actin filaments, and the big black nucleus in the center. This is done with film on a 120 kV electron microscope, and you would be hard-pressed to get a picture as good as this in today's modern electron microscopes. So the work that these people did back in the 50s is really incredible for the technical abilities that they had. So biological electron microscopy requires careful sample preparation. Every step that you take is important to the next step that you take. Biology studies living things. We want to look at the cells that we prepare in a state that they were when they were alive, all right, under conditions of high vacuum and when they're being bombarded by electrons, which are not conducive to life. So we have to be very careful that we're not looking at a dead plant, but we're looking at a live, functioning, alive plant. All right, so to get to this final goal of looking at something as it was when it was alive, we're going to have to define that goal very clearly in our scientific protocols. Preparation of TEM specimen is specific to the material under analysis. So if you're looking at a monolayer of cells, if you're looking at tissue, if you're looking at purified proteins, the preparation is going to be different depending on what you're looking at. If you're looking at yeast versus mammalian tissue, preparations are going to be a bit different. And also depending on the questions that you're asking. If you're wanting to look at ultrastructural morphology of liver, you're going to prepare that tissue differently than if you're looking at purified proteins, for example. So you need to define your goals, define your questions before we get going. We also want to plan, again, your experiment very carefully. You want to define your question. I often get questions like, well, I just want to see what it looks like. which is much, much too vague of a question to ever consider answering with an electron microscope. You need to say, I want to know what the mitochondria look like under conditions A, B, and C, comparing that to conditions of purity and absolute healthy state of minds. So plan your question, define your question specifically, plan carefully. You want to have proper controls. Think about the ultimate end result that you're going to get and understand that biology is a diverse field. So cells within the same culture plate are going to look different from each other. They're going to be slightly different. So you've got to plan for those controls and think about how, if it's a little bit different, how am I going to know this is the experimental versus the healthy normal control? We prefer not to look for a needle in a haystack. If you're transfecting cells, for example, we don't want to look at a transfection rate of 10% in your culture medium. It's going to be very difficult to find those cells once we've pelleted and sectioned them. If you look in a section and you have 100 sections of cells, only one of them would be transfected out of 100. That's not very good sampling, right? You're not going to be able to draw great conclusions unless your needle is very big. So if you're looking for a needle in a haystack, you better make sure that that needle is very, very big, or the sample is very small. So you want it to be an unmistakable result of what it is that you've done, if it's only going to happen once out of 100 times. So the primary methods that we're using in bio-TEM is negative staining, routine morphology. We could do protein localizations with immunolabeling. And now with cryo-TEM, we can start looking at nanostructures of purified proteins. Negative staining, we'll start there. It's a fast, easy, quantitative method of examining structures, isolated molecules, small cells, or purified organelles. If we think of this as an electron transparent sample that you've purified and you've surrounded it by an electron dense stain, and it appears in negative contrast, as opposed to the object itself being stained and the background being white, which would be positively stained. It's always going to be your first step if you're going to cryo-EM or single particle analysis. If you look at this micrograph here, this is a negative stained micrograph of purified proteins. You can see that we can have really, really high level resolution detail here. We know that this prep is clean. It's of a good concentration level. The objects are orienting themselves in infinite orientations so that we can do later a 3D reconstructions of this protein, or we may even find that we've got all the answers we need right now. And we don't have to stand in line for a cryo Titan electron microscope for 6 months to get a better look at this. So negative staining is always your first step before you start going on to cryo. The challenges you're going to run into are what you're going to run into with any sort of sample prep. You need to have a monodispersed layer of particles. If they're laying on top of each other, you're not going to get resolution of a single particle. You need to worry about your compatible buffers, compatible with the stain you're using. Some buffers are going to precipitate stain, some won't. HEPIs is good, PBS is good, or if you can, water is probably your best bet. Structural artifacts are common with negative staining, and that's why cryo was invented, frankly, such as flattening of spherical structures. The sample is air dried, and there's your problem as well. If you're wanting to look at something in a native state, air drying is not where you want to be. Sample concentration is critical. Cleanliness is critical. Purity is critical. And the stain, because it's binding to your structure, is going to interfere with resolutions of fine structures. When you start magnifying things, you get to a point where you're just magnifying the stain. The benefits is it's very high contrast, it's fast, it's easy, sample prep, and it's easy for anyone to master. So if you have a lot of conditions that you're trying to get through, you can get through 30, 40 conditions in a week with negative staining. The method, again, is quite simple. We start with their grid preparation. Usually we're using copper grids because, again, they're cheap, easy, and reliable. 200 to 400 mesh grid. So that's the screen size on the grid. We're going to have a carbon substrate on top so that our sample doesn't fall through the holes. Sometimes just carbon, sometimes carbon formvar. And we're going to freshly ionize or glow discharge the grids so that we have a hydrophilic surface that allows a more even distribution of your sample and a more uniform staining when we're done. The method simply is you purify your protein, you absorb it onto a grid, you either drop cast it onto the grid or you float your grid on top of a drop of the sample. Pick it up, you can dry the sample sometimes, or generally you just wash it when it's wet with 5 drops of uranyl acetate. Then you dry that down onto your sample. Don't wash again. Let the stain dry down so it's surrounding your sample. Most common stains that we're using, 1 to 2% aqueous uranyl acetate, phosphotungsten acid, or 1 to 2% ammonium mobylate. They all have different pHs, and so that may affect and interact with your buffers or with your proteins differently, creating better or worse samples. It's not likely that you're going to have a situation where one stain will work and the others will show nothing. Usually you're getting a situation where 2% aqueous solution works, but you'd see if you can get a little bit better resolution, so you might try another stain. In a core facility, what I'm going to provide is 1 to 2% aqueous UA because it has a long shelf life and it works for 90% of the samples that we're looking at. If we wanted to try something else, we can talk about it, mix it up. Some of these have shorter shelf lives, have to be made up immediately before use. So it's not practical for me to keep them actually on hand. But we can get really good pictures. Again, we've got some latex nanoparticles on the right. We see no structural damage here. There might be some aggregation going on. We don't know if that's because they actually do aggregate or if it's because of sample. We can't tell that by looking at it. On the left is regular lipid nanoparticles, and you can see that there is some structural damage with those particles as they've dried down. The structure has collapsed a little bit. You think of it as like a collapsed beach ball, and stain collecting in those wrinkles of the collapsed particle. But you can still gain a lot of information that you're going to need before going to cryo. You've got to clean samples, evenly, homogeneously dispersed, and you could do that with different samples. Yeah.
What's the scale bar? Your nano.
I don't know. I'm sorry. They're old pictures that I just grabbed out of my file.
So your nanoparticle inside is your target, outside is the nanoparticle, or you say that is
This is probably artifact, this white background, and then these are the particles.
okay.
And they're probably they're probably 10 to 15 nanometers diameter.
We used to like carbon substrate sometimes. Carbon grades will cause this problem background.
What was the background?
Carbon grades.
Oh,
Can cause this problem.
yeah, you're right. Yeah. Sometimes you get that particular background. Yeah, that's good to know. You can also look at whole cells. Here's a whole bacteria cell. Again, by looking at this, we don't know if the wrinkles or ruffles on the surface of that bacteria are real. But we can see through it. We can see the outline of the cell. We can measure it. We can do other questions. If we wanted to know if the wrinkles were real, we'd probably have to embed and section it. We were concerned more about the extracellular vesicles that the bacteria was releasing, so we didn't care so much about the bacteria itself. But you can see, you can get really good pictures just even of something as large as a bacteria. All right, so if we move on to classical EM or ultrastructural morphology, we're going to be looking at tissue, cells, bioengineered complexes, things along this way. It's considered classical EM. It consists of ultra-thin sections of plastic embedded material. So we have a much more involved process of sample production involving fixation, contrast enhancement, dehydration, embedding, sectioning, staining, scoping. So this is going to take about a week or two to accomplish as opposed to minutes to complete. We're generally asking questions about organelle distribution. We're characterizing an effect from a transfection or a drug treatment, or looking at morphometry, where we're counting how many incidences of occurrence may happen in a population of cells. The problems we're trying to overcome is that in a whole live cell, we have an 80% water in this cell. That's not going to work in a vacuum. And the vacuum is going to just be destroyed if it's not treated in some manner. We're also only looking at a section. So we have to be careful to not overinterpret or underinterpret the results that we're getting, because we're just looking at a fraction of the cell. Population of the cells in your pellet comes into play here. So if you have enough cells in your section, you can hope or assume that they're all cut at different planes. And so you could reconstruct, if you wanted to, a whole cell based on the information that it's a small section in each part. And also biological material is basically transparent. The only reason we can see each other is because we're thick enough, right? Otherwise we're invisible. So we're going to have to do some sort of contrast enhancement. So the steps that we're going to do that, first is fixation. We're going to use glutaraldehyde as our fixation, as two aldehyde groups, so we get good cross-linking. We're also going to throw in a little bit of paraformaldehyde, because paraformaldehyde is a small molecule, can penetrate quickly into the cell and fix quickly, and then the glutaraldehyde follows behind and does the cross-linking. We're going to wash that and then stain it with contrast enhancements. We're going to use osmium tetroxide, and we're going to follow that by uranyl acetate. After that, we're going to wash quickly and dehydrate it completely, replace all the water in the cell with ethanol. We're going to do that slowly so that we don't shrink or explode the cell in that process. So we start gradually, 70%, 80, 90, 95%, multiple changes of 100%. This is where you may alter your protocol a little bit, depending on what it is you're actually embedding. Bacteria, yeast, plant material, things with cell walls are going to have a harder time to dehydrate. They're basically designed to not dehydrate. So longer periods of time, more changes, things along those lines would be an adaptation you might make. Then we're going to go into propylene oxide, which is a nice carrier solution for our liquid plastic or liquid resins. And then multiple changes of 100% resin, so that we have full, complete resin has now replaced all the water. So we replace the water with ethanol, ethanol with propylene oxide with resin. Now we put the resin into a mold, we put our tissue into that mold, polymerize it at 60 degrees, and we have a very, very hard block. The hard block is what allows us to then section very, very thin. At this point now we're sectioning, we've got our hard block, black piece of tissue at the tip of the block, trim that very carefully, face it so we've got a mirror smooth finish. And then we finally put on our good diamond knife and cut ultra thin sections. After that, we're also going to stain it again for more contrast enhancement of all the fine membranes. All right, so concerns and problems. Routine morphology sample prep is not hard. It's simply changing solution after solution, but it's unforgiving. The problem, what makes it hard, is that you can't make any mistakes. If you have made your fixation improperly, or you have slow fixation, then you don't get good membrane preservation. And the membranes is basically what's defining all of our organelles. So you have no contrast then. If you have incomplete dehydration, results in holes in the final block, which makes it very, very difficult to cut and image the sample. If you have an incomplete resin change and you leave some solvents behind, then as you start sectioning and cutting it onto the water surface of the diamond knife, that's just going to dissolve on that surface. If you've formulated your plastic improperly. When you made your plastic, you may end up in a block that's too soft or it's gummy, or actually it can be too hard. And so when you go to cut it, it just shatters like crystal instead of cutting like a nice plastic block. Dull, damaged knives are going to result in the block face being covered with scratches, which makes imaging impossible as well. And then when you're finally all done with all of that, you've done all that right, and now you go to stain it for a final time, your lead or your UA can precipitate all over the sections and you have to start over with sectioning. Or the lead just doesn't stain at all. And so you've done all this and you still have no contrast. So there's all sorts of things that are going to trip you up that you need to keep track of and control for. So in CNS, just to take a moment here, if you're interested in this sort of imaging, we could do training or service. And there's basically 3 options for routine morphology. The users can be trained to competency from the entire step of the process, where you do the embedding, the sectioning, and finally the imaging. Or if you wanted to not spend your time learning how to embed and section, which is a time commitment. It's not, sectioning is not an easy thing to do at a publication quality level, then that can be done as a service for you and you're trained on the TEM. Or we could just work together and I could do the entire process. If I'm going to do the entire process, I need to know more about your work so that when I'm imaging for you, I can do it with some purpose and not just randomly take pictures. Yeah.
We have some cheese sample.
Cheese, teeth.
Yeah.
Yeah.
Only some.
I don't know. I've had conversations with about teeth before. Teeth I would treat like bone, and typically bone needs to be decalcified
Yes.
before it can be embedded. I don't know if that would work with teeth because of the enamel.
When to make artificial teeth, so we really wanted to look inside the nanostructure.
Yeah, of teeth. We'd have to do, I have not ever done that. So I'll be honest from the start, I don't have a, yep, do this, do this, do this. I'm sure it's been done and that protocols can be found. And then we can read through them and see if that fits your experimental protocol or not. Yeah, yeah. I don't think it's going to be easy because teeth are hard. It's bone, so you can't just cut it. Yeah.
Very.
Yeah, and it's big, so embedding it makes it hard too.
SEM maybe first.
You might want to start there.
Thank you.
Yeah. Okay. So now on to cryo-EM. Everybody wants to do cryo-EM. I like to show this picture of the ribosome because it heralds back to George Pilates' day, and it's an honor to him, I think, that people still are working on the ribosome all these years later. So what are you looking at with cryo-EM? We're looking at whole cells. If they're thin enough, we can get virus particles, proteins. You can do tomography. You can do single particle. All of this is in the realm of cryo-EM. But why are you doing cryo-EM? Why? Not just because it's going to get you published, and not just because it's the hot thing to do right now, but why? And the reason why you're doing cryo-EM is because you're wanting to look at hydrated, unstained samples. When you air dry samples, when you air dry proteins, they change, potentially. So looking at them hydrated is how you're going to look at them in a more natural state, as opposed to the air dried negative state. Other than that, there's nothing magic going on here. The sample is never dried. The sample has no contact with the surfaces, so that's going to possibly preserve orientations that might be interesting. And there's no stain present to obstruct structures or small crevices. So high resolution images of 3D molecules, single molecules in 3D, and allows possible examination of dynamic or functional states. If you can possibly freeze your proteins in different functional states, conformational changes, then that can give you a lot of information too. All right. So as I said, the sample is more like a native state. We want to look at things as they are fresh and alive, and the flash frozen peas is going to be more like the fresh alive peas. The negative staining is going to be more like canned peas. Still good, still fine, but maybe not quite as good as brand new, fresh off the trees. And if you're used to studying proteins with Western gels, well, that's where you get, that's like your pea soup. So that's a long ways away from the fresh and alive peas. So we're looking at things in more of their native state. So cryo-EM allows imaging of macromolecule structures to the atomic resolution. The detailed structural models obtained from reconstructions grant insight into the function of macromolecules and their roles in the biological process. Pharmaceutical organizations are using cryo-EM to analyze ligands, ligand bindings, perform conformational studies, and carry out drug testings that can be exploited for new drugs and disease model corrections. Cryo-electron microscopes over the past 10 years have become more amenable to automation through features like autoloaders and mechanical stages that decrease drift and aberration corrections. So now what the problem still comes down to the same problem we've always had since Dr. Pilates' day. Sample preparation is the main impediment to workflow. Once you get your sample prep done, you're moving forward. And why is sample prep so hard? Because we're freezing samples, preferably in water. Water has different states. We're taking liquid water and we're freezing it. What we want is vitreous ice with 0 crystals. What we often get is hexagonal cubic ice. That's fantastic for skiers. That's what you want, that frosty powder that you want to ski on. But that's no good if you're doing EM. So that's where the problem lies. It seems like an easy problem to solve. But when you're talking about minus 40 degrees Celsius, fingers and things freeze. So things just get harder. All right, so what do we need to think about? We need to first optimize your sample with negative staining. We've shown that to be true. We can make a lot of progress on sample prep by just optimizing with negative staining. And now you don't have to test 10,000 conditions by cryo. Grid preparation, this is your first hurdle to overcome. You do want to ionize, again, to make it a cleaner, better surface for your proteins to adhere to. But now do you choose gold or copper? Is your protein going to behave differently on gold or copper? It might. Mesh size. You can take 200 mesh, 300 mesh, 400 mesh. Which one is going to be best for your sample? It might make a difference. The hole size, because we're freezing, these grids are made up of a carbon film with uniform holes in it. That hole size may make a difference as well. The ways it may make a difference is that you may have all of your proteins freezing in one orientation. So now you're not getting a 3-dimensional structure, you're just seeing one orientation of the structure. So that's one thing to be concerned about. There's many variables that are encountered in a sample and grid preparations that make it difficult to identify a cause and effect relationship. We can't say, if you have A, then do B. There's so many factors that are being involved here that it's difficult to have a linear progression of events. So while you're preparing a grid itself does not take a lot of time, the number of iterations that you might have to go through to get the perfect sample does take a lot of time. And that's why it's difficult to standardize these protocols. Unfortunately, we're in the stage of cryo-EM's development as a methodology. We're all still learning. Everybody's going to have to sort of make their own protocols for their own proteins. And hopefully when you're publishing your papers, you're making good notes in your methods that they can really build on this body of knowledge that we're creating right now, because it's still in its infancy. So be clear and very concise about what you've done and why you did it. So, but the basic sample prep is Again, negative staining. I can't say it often enough. Start with negative staining. And then determine your optimal freezing conditions. What we're going to do is we're going to put sample on a grid. We're going to blot that grid to a particular thickness, and then we're going to drop that quickly into liquid ethane that's at liquid nitrogen temperature, so minus 190. Hopefully what you're going to end up with is a monolayer of protein in a monolayer of ice that has 0 ice crystals or vitrified ice. The picture on the side is the Vitrobot, which is the standard instrument used for plunge freezing. Leica has a equivalent instrument now as well. And there are other instruments coming along, jet spray freezing, other ways of that people are trying to find to make this part of this process a little bit easier. It's just sort of test them all if you're in the market for buying one of these things until you find what works nicely in your hands or that works nicely with your modes of study. And then what we have then in the bottom there is just the grids. You can see the holes and the sample in the hole. All right. When we're imaging their sample, we're using the scope in low dose. So we're basically setting up the microscope so that we can focus and examine one area and then shift the beam so that we are now taking a picture in an area that's never been looked at. We do suffer a great beam damage with frozen samples. You've got a hot electron beam. It's going to burn up the sample. If you look at it too long, it's just going to boil the sample. And also we have a low signal to noise. To keep in mind, you're going to have to do some, post-acquisition analysis of your images. You're not going to be able to look at the scope and determine anything. You're just going to be taking automated pictures. And then also what you need to keep in mind with your sample preparation is that you're going to have millions of pictures of your object to do a really high resolution 3D reconstruction. The 3D, the resolution comes from averaging of millions and millions of objects of the same thing. So we have very low contrast, as I said. The early days of cryo, they talked about under focusing to get better contrast. Now we don't doing this so much because you're starting to affect, if you're looking for angstrom level resolution, under focusing is going to negatively affect that ability to get high level, angstrom level resolution.
So one was the substrate is carbon grade, possibly UV grade, right? But just
I'm sorry.
the substrate. The carbon substrate.
Yes. Yes.
Purchase.
Yes. The user would purchase this. Are you referring to like the lag time and being able to buy them? Yeah. What I've just found out is that Quantifoil, who is the primary supplier, they've had this big lag time. So it's like 6 to 9 months or longer. But they are opening their new factory supposedly in September, which is going to cut that lag time way, way down. So that's the scuttlebutt. But also many of these other companies are making their own grids. So you don't have to necessarily go to Quantifoil. They all have their own grids that they do have in stock. So you might want to consider buying something else. Yeah, it's a problem. So then we get the single particle 3D reconstruction. We've got herpes virus, one of the first single particles that were solved. We get a 3D model out of 2D images. Micrographs are collected with or without tilt. Objects are selected, aligned, classified. Thousands of objects are needed to reconstruct a three-dimensional model. And then the more modern up-to-date, where you have software that can actually perform some of these tasks for you. First, you're collecting, again, all the images with thousands of millions of objects in them. You're using a software to categorize them, weigh them, throw out good ones, keep bad ones, or throw out bad ones, keep good ones. And then finally, rebuild a model with all the iterations that you need to until you get the higher level resolution and fit that back to your old model of what your protein should look like. Disadvantages of cryo-TEM. Well, one thing I didn't put on here, the microscopes are very expensive, so that's probably barrier number one. But it's a challenging technique to learn. It takes time and patience. And if you've never done any EM before, you might not, you just be aware it's going to take some time and some practice. All right. Specimen concentration needs to be high. Samples are going to be very sensitive to beam damage. The images are very low contrast. Vitreous ice is not easy to form. You're going to have to find the right concentrations of buffers and right concentrations of samples, the right amount of blot time, the length of blot time, the force of blot time, the number of blots. All of these things are going to play into the quality of your vitreous ice when you're done. And if you can't get vitreous ice, ice crystals are going to damage the sample basically and make imaging impossible. So you have to master it. There's no option. Just for references, if you're new to cryo-TEM, there's a good paper in Nature Methods, Understanding Sample Preparation for cryo-EM. And if you are going to be doing any cryo-EM analysis, you really should look up and watch the Caltech course concerning analysis of cryo-TEM images. It doesn't go into so much of the sample preparation and acquisition as it does the analysis after the images are taken. So it's good to have all that math in your hand and in your head when you're starting to take these images. All right, so just in conclusion, the most difficult aspects of biological electron microscopy is the sample preparation of the sample material. Once a number of basic techniques have been mastered, the beginner can be trained quickly to operate the electron microscope for biological purposes. You've seen other talks this summer series where you're doing amazing things with EDS and electron detectors and these sorts of things. For us biologists, we're really not using the microscope to its full capacity in that manner. We're really using it as a camera to document what we've done. So define your question, choose the correct preparation technique, and determine your controls, and never forget your controls, and never forget your negative controls. You want to know what perfect looks like in your lab, in your incubator, in your hands. All right, so I just like to also add as a plug, bio-EM, is kind of sometimes falling out of fashion, especially cryo is taking over, but there's few biological research problems which do not benefit from the use of some electron microscopy. If you're doing cell biology, you should be doing electron microscopy. So, and if you're interested in including EM analysis in your research project, please contact me for a consultation, and we can talk through whether it's a good project or a reasonable project or something that can be done. All right. And when we do talk, send me the supporting materials so that I can understand what you're talking about. Right. And with that, I'll just say thank you. Questions?
I do have one more question.
Okay.
How you really avoid the beam damage in the sun, which is a really kind of circle. The EV damages the sun.
Oh,
Really needed to be sun.
yes, but it's inevitable. I mean, you can't avoid beam damage on a sample. You can, but then you might not have enough power to get through the sample. So then your sample prep in terms of freezing is going to come into play where you have room to damage. If something's going to be easily damaged, I'll sacrifice one if I have 10 more. So that's kind of the strategy. It's just have backups. Yeah. I mean, if it's going to get damaged, it's inevitable. And some things damage faster than others. Even a negative staining, you can throw just normal 80 kV, 100 kV on negative staining, and it starts. And that's basically sample dependent. I mean, you like to think that it's stain dependent or something, but it's sample dependent. It's always sample dependent. Yep. How do we know if it's?
Our students, do you have any question? I love to let you ask a question.
Yes. Yeah.
Thank you.
Okay.
Good.
Good.
Thank you so much.
You're welcome. Thank you.